RNA splicing is a fundamental process in molecular biology where the cell edits genetic information. This process involves removing introns, which are non-coding sequences, and keeping exons, which are coding sequences. Think of it like editing a rough draft of a document by cutting out unnecessary parts and keeping only the essential information that will be used to create proteins. Splicing is a critical RNA processing step that occurs after a gene has been transcribed into pre-messenger RNA. During this step, the cell identifies and removes non-coding sequences called introns from the pre-messenger RNA molecule. At the same time, it joins together the coding sequences called exons to form mature messenger RNA. This mature messenger RNA is the final product that will leave the nucleus and be translated into proteins. There are two key players in the splicing process. First, we have introns, which are non-coding sequences that do not contain instructions for making proteins. These sequences are removed during splicing and discarded. Second, we have exons, which are coding sequences that contain the actual genetic instructions needed to build proteins. These exons are kept and joined together to create the final messenger RNA molecule that will be translated. Before splicing occurs, the pre-messenger RNA has a specific structure. Starting from the 5' end and moving to the 3' end, the molecule alternates between exons and introns. For example, you might see exon 1, followed by an intron, then exon 2, another intron, and finally exon 3. This alternating pattern is typical of many genes in eukaryotic organisms like humans, animals, and plants. During the splicing process, the pre-messenger RNA undergoes a dramatic transformation. The molecular machinery identifies all the introns in the molecule and cuts them out precisely. The remaining exons are then joined together in the correct order. So our pre-messenger RNA that had exon 1, intron, exon 2, intron, and exon 3 becomes mature messenger RNA with just exon 1, exon 2, and exon 3 connected seamlessly. This mature messenger RNA is now ready for the next step in protein synthesis. The molecular machine responsible for splicing is called the Spliceosome. This is a large complex made up of small nuclear RNAs and proteins that work together with remarkable precision. The Spliceosome has three main jobs. First, it recognizes the specific sites where introns and exons meet, called splice sites. Second, it positions itself correctly at these boundaries. Third, it catalyzes the chemical reactions that cut the RNA at the intron-exon junctions and join the exons together. This entire process must be extremely accurate because even a single mistake could result in a non-functional protein. Splicing is incredibly important for several reasons. First, it removes non-coding sequences from the RNA, ensuring that the genetic instructions are accurate and contain only the information needed to build proteins. Second, splicing allows for exon shuffling, a process where different combinations of exons can be joined together. This creates diversity and allows a single gene to produce multiple different protein variants. Third, splicing prepares the messenger RNA for translation, the process where ribosomes read the messenger RNA and synthesize proteins based on its instructions. To summarize, splicing is the precise editing of RNA transcripts. The cell removes introns, which are non-coding sequences that do not contribute to protein structure, and joins together exons, which are coding sequences that contain the genetic blueprint for proteins. This process creates functional mature messenger RNA that is ready for translation. Understanding splicing is essential for comprehending how genetic information flows from DNA to RNA to protein in living cells.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.